Hey guys, this presentation is on the neuromuscular junction and excitation-contraction coupling. The peripheral nervous system has two motor divisions. The somatic motor system controls voluntary movement by targeting muscle cells, and the visceral or automatic motor system regulates involuntary responses in internal organs. Efferent neurons from the somatic motor system travel along cranial or spinal nerves and synapse with skeletal muscle cells. These structures are called neuromuscular junctions. Again, muscle contraction consists of a motor neuron labeled here as number 1 and axon terminals labeled here as number 2. The axon terminals interact with the muscle fiber, which is labeled as number 3. The neuromuscular junction occurs between numbers 2 and 3, so let's take a closer look. A neuromuscular junction is defined as a chemical synapse between a somatic motor neuron and a skeletal muscle cell. The neuromuscular junction is what allows the electrical signal sent by the motor neuron to be translated into neurotransmitters. Once the signal is translated, the skeletal muscle cell receives the signal and becomes excited. This results in contraction. Let's go through the first steps of this process at the neuromuscular junction. First, we'll label all of the important structures of the neuromuscular junction. This structure here, labeled as A, is the somatic motor neuron. The part of the structure labeled as B is the axon terminal of the somatic motor neuron. This is the part of the motor neuron that releases the neurotransmitters in order to relay signals across the synaptic cleft. At the axon terminal, there are many synaptic vesicles filled with neurotransmitters called acetylcholine. Here they are shown as white circles with purple dots in them. The purple dots are the acetylcholine neurotransmitters, and the white circles are the vesicles that hold these neurotransmitters. The gray stripes shown on the membrane of the axon terminal are calcium voltage-gated channels. These channels allow calcium into and out of the motor neuron. Here, the skeletal muscle cell is labeled as D. The purple portion of the muscle cell is the sarcolemma. This part of the muscle cell is what interacts with the motor neuron, and it is also known as the motor end plate. This part of the muscle cell is folded in order to create more surface area. More surface area allows for a higher concentration of chemical-gated cation channels called acetylcholine receptors. These are shown as the blue stripes on the sarcolemma. The synaptic cleft is the space between the motor neuron and the skeletal muscle cell. Here it is labeled as C. Now that we have labeled all the important structures, let's look at how the neuromuscular junction works. Synaptic transmission in the neuromuscular junction all starts with an action potential. When the axon terminal of a motor neuron receives an action potential, calcium voltage-gated channels will begin to regulate the influx of calcium ions into the neuron. As the calcium, shown here as the red dots, enters the cell, they will try to bind to synaptic vesicles. When the calcium binds to the synaptic vesicles, the acetylcholine neurotransmitters inside of them will be released. When the acetylcholine neurotransmitters have found their way out of the motor neuron and into the synaptic cleft, they bind to acetylcholine receptors on the motor end plate. When acetylcholine neurotransmitters bind, acetylcholine receptors will open to allow sodium ions, shown here as the yellow dots, into the skeletal muscle cell, and potassium ions, shown here as the blue dots, out of the muscle cell. Due to the fact that more sodium ions diffuse into the muscle cell than potassium ions out of the muscle cell, the net difference of the cell is positive. This positive difference will cause an end plate potential. An end plate potential causes the depolarization of the muscle cell, which can trigger another action potential, and because this occurs at the motor end plate, this second action potential will spread across the entire sarcolemma. When this happens, it is the beginning of excitation-contraction coupling of the muscle cell. Excitation-contraction coupling is a series of events triggered by an action potential in the sarcolemma of the muscle cell. In order for the skeletal muscle cell to begin excitation-contraction coupling, the membrane needs to be stimulated by an action potential so that it will depolarize and trigger another action potential. This would be the excitation part of excitation-contraction coupling. The excitation of the muscle cell begins when acetylcholine receptors open to allow positively charged ions into the skeletal muscle cell. As depolarization occurs, voltage-gated sodium channels in the sarcolemma are triggered to open. Sodium ions enter the muscle cell through the sodium channels, and as this is happening, the action potential spreads across the entire sarcolemma of the muscle cell. The action potential is able to spread by sequential opening and closing of voltage-gated sodium and potassium channels. This is propagation of the action potential, and it occurs just as we previously learned. Propagation of the action potential along the sarcolemma is the excitation part of excitation-contraction coupling. Invaginations in the sarcolemma, called T-tubules, carry the action potential from the surface of the cell to the interior of the skeletal muscle cell. The action potential triggers the opening of calcium channels located in the membrane of the sarcoplasmic reticulum. A triad is the arrangement of a T-tubule with membranes of sarcoplasmic reticulum on each side. A triad surrounds the myofibril, shown here as the blue cylinder, which contains sarcomeric proteins actin and myosin. As calcium diffuses out of the sarcoplasmic reticulum and into the sarcoplasm, contraction of the muscle fiber is initiated. The change in sarcomeric proteins, shown here as thick filaments and thin filaments, in response to the influx of calcium, is the contraction portion of excitation-contraction coupling. The cross-bridge cycle describes the change in sarcomeric proteins that occurs during the contraction of the muscle fiber. Should there be any problems with these sarcomeric proteins, the contraction of the muscle would not occur correctly. If, for some reason, any sarcomeric protein was absent, the thick and thin filaments would not be able to generate movement together to create muscle contraction. Therefore, the muscle would be excited by all of the previous steps, but it would never contract. If there was too much of a protein, this would cause the muscle cell to contract constantly and cause muscle fatigue. Thank you for watching my video, and I hope I was able to help you understand the process that occurs at the neuromuscular junction as well as the beginning of excitation-contraction coupling. All images used in this presentation came from the following references.